Aptamer therapeutics refer to a class of therapeutic agents that are based on aptamers. Aptamers are short, single-stranded DNA or RNA molecules that can bind to specific target molecules with high affinity and specificity. They are often referred to as chemical antibodies due to their ability to recognize and bind to target molecules, such as proteins, peptides, small molecules, or even cells. Aptamer therapeutics have gained significant attention in the field of medicine and biotechnology due to their unique properties. They can be designed and selected to bind to a wide range of targets, including disease-associated proteins or receptors. Once bound to their target, aptamers can interfere with biological processes, such as blocking protein-protein interactions, inhibiting enzymatic activity, or modulating signaling pathways. The potential advantages of aptamer therapeutics include their high specificity, low immunogenicity, and ease of synthesis and modification. They can be chemically synthesized, allowing for the incorporation of various modifications to enhance stability, pharmacokinetics, and target binding affinity. Aptamer therapeutics have shown promise in various applications, including cancer therapy, targeted drug delivery, diagnostics, and regenerative medicine. They offer a versatile platform for the development of novel therapeutics, with potential benefits in terms of efficacy, safety, and personalized medicine. However, it is important to note that aptamer therapeutics are still in the early stages of development, and further research and clinical trials are needed to fully explore their potential and establish their efficacy and safety profiles. Using aptamers for therapeutic applications offers several advantages. High specificity. Aptamers are single-stranded DNA or RNA molecules that can bind to specific target molecules with high affinity and specificity. This allows for precise targeting of disease-related proteins or biomarkers, minimizing off-target effects. And versatility. Aptamers can be designed to target a wide range of molecules, including proteins, peptides, small molecules, and even whole cells. This versatility makes them suitable for various therapeutic applications. 9. Low immunogenicity. Aptamers are chemically synthesized molecules, which reduces the risk of immunogenicity compared to biological therapeutics like antibodies. This makes them potentially safer for use in patients. Ease of synthesis and modification. Aptamers can be synthesized using automated processes, allowing for efficient production and scalability. They can also be easily modified with different functional groups or conjugated to other molecules, enabling the delivery of therapeutic payloads or imaging agents. Stability and shelf life. Aptamers can exhibit excellent stability, both in terms of storage and in biological environments. They are less prone to degradation by enzymes or harsh conditions, ensuring a longer shelf life and improved therapeutic efficacy. Reversible binding. Unlike irreversible inhibitors, aptamers can bind to their targets reversibly. This property allows for dynamic regulation of target activity, making them suitable for applications where on-off switching is desired. Lower production costs. Aptamers can be synthesized using chemical methods, which can be more cost-effective compared to the production of antibodies or other biologics. This makes aptamers a potentially more affordable therapeutic option. These advantages make aptamers a promising class of molecules for therapeutic applications, including targeted drug delivery, diagnostics, and personalized medicine. One common use of aptamers in synthetic biology is as biosensors. By incorporating aptamers into a detection system, they can selectively bind to a target molecule and trigger a signal, such as fluorescence or color change, indicating the presence or concentration of the target. This enables the development of sensitive and specific detection methods for various analytes, including toxins, pathogens, or disease biomarkers. Aptamers can also be used as molecular switches or regulators in synthetic biological circuits. By designing aptamers that undergo conformational changes upon binding to a specific molecule, they can be integrated into genetic circuits to control gene expression or cellular behavior. This allows for precise control of biological processes and the creation of programmable systems. Furthermore, aptamers can be utilized in drug delivery systems. By conjugating aptamers to therapeutic molecules, they can selectively target specific cells or tissues, enhancing the delivery and efficacy of drugs. This targeted approach minimizes off-target effects and improves the overall therapeutic outcome. Overall, aptamers play a crucial role in synthetic biology by providing versatile tools for molecular recognition, biosensing, gene regulation, and targeted drug delivery. Aptamers have several potential uses for in vivo applications, where they can be utilized for various purposes. To add to therapeutic applications, aptamers can be developed as therapeutic agents to target specific disease-related molecules. They can bind to disease-associated proteins such as receptors or enzymes and interfere with their function. This approach can be used for targeted therapy, where aptamers deliver drugs or inhibit the activity of specific proteins involved in diseases like cancer, viral infections, or inflammatory disorders. Aptamers are a fascinating class of molecules widely used in therapeutic applications due to their unique ability to bind specifically to a variety of molecular targets. To understand the biochemistry and molecular biology of aptamers in therapeutic applications, we need to cover several key areas. Nature and synthesis of aptamers. Aptamers are single-stranded DNA or RNA molecules that can fold into unique three-dimensional structures, capable of binding tightly and specifically to target molecules, proteins, small molecules, and even cells. The process of creating aptamers typically involves a method called systematic evolution of ligands by exponential enrichment, SELEX, which selects molecules that bind with high affinity to a target from a vast library of random sequences. Binding mechanisms. The binding specificity and affinity of aptamers to their targets are akin to antibodies, making them excellent for therapeutic use. This specificity comes from their ability to form complex spatial structures that can fit perfectly with a specific part of the target molecule, similar to how a key fits into a lock. Modifications and stability. Aptamers are often modified chemically to increase their stability and resistance to degradation by nucleases in the body. Common modifications include the addition of non-natural bases, backbone modifications, like replacing phosphodiester bonds with phosphorothioate bonds, and the addition of molecules such as polyethylene glycol, PEG, to increase their size and circulating half-life. Therapeutic applications. Targeted drug delivery. Aptamers can be used to deliver drugs directly to specific cells or tissues, reducing side effects and improving drug efficacy. Diagnostics. Due to their high specificity, aptamers can be used in diagnostic assays to detect the presence of proteins or other biomarkers associated with disease. Regulation of biological pathways. Aptamers can act as inhibitors or activators of specific proteins, thus regulating cellular pathways that are critical in diseases like cancer or autoimmune disorders. Neurodegenerative diseases. Aptamers can cross the blood-brain barrier, a rare feature that is highly valuable in treating diseases such as Alzheimer's or Parkinson's by targeting brain-specific proteins. Clinical development and challenges. Despite their potential, the path from bench to bedside for aptamers involves significant challenges. These include issues of toxicity, immune response, and manufacturing costs. However, some aptamers have reached the market or are in late-stage clinical trials, proving the utility of this approach. Examples of therapeutic aptamers. Pegaptenib, Macugen. Used to treat age-related macular degeneration, this aptamer blocks vascular endothelial growth factor, VEGF, which is responsible for the growth of abnormal blood vessels in the retina. Nusinersen, Spinraza. Although not strictly an aptamer, this antisense oligonucleotide is similar in concept and is used to treat spinal muscular atrophy by modulating the splicing of the SMN2 gene. Diagnostic tools. Aptamers can be employed as diagnostic agents for in vivo imaging or detection of specific molecules. By conjugating aptamers with imaging agents, such as fluorescent dyes or nanoparticles, they can selectively bind to disease markers or targets of interest. This enables the visualization and detection of specific molecules in vivo, aiding in disease diagnosis and monitoring. Aptamer modifications for enhanced diagnostic functionality. For diagnostic purposes, aptamers often undergo various chemical modifications to enhance their stability, binding affinity, and resistance to enzymatic degradation. Modifications can include locked nucleic acids, LNAs, enhance hybridization properties and increase resistance to nuclease degradation. 2. Fluoro and 2. O-methyl modifications. Improve nuclease resistance and affinity. Biotinylation or conjugation with fluorophores. Facilitate detection and signaling in assay systems. Applications in diagnostics. Biosensors. Aptamers are used in biosensor devices to detect the presence of specific analytes by causing a detectable change in the sensor's output upon binding. ELONA, enzyme-linked oligonucleotide assay. Similar to ELISA, but uses aptamers instead of antibodies for the detection of proteins or other molecules, which can be quantified after an enzyme reaction produces a color change. Molecular imaging. Aptamers labeled with radioactive or fluorescent tags can bind to specific cellular targets, allowing for imaging of tissues and identification of diseases like cancers or inflammatory processes. Advantages over other biorecognition elements. Temperature stability. Aptamers do not denature at high temperatures, unlike proteins. Astronaut. Synthetic accessibility. They can be chemically synthesized with high reproducibility. Versatility. They can be engineered to have reversible binding characteristics. Challenges in diagnostic applications. 8. Regeneration and reuse. In biosensing applications, it's often necessary to regenerate the aptamer to reuse the sensor, which can be challenging depending on the binding strength and the nature of the target. Sensitivity and specificity. While aptamers are generally highly specific, achieving the necessary sensitivity for certain low-abundance targets can be challenging and may require signal amplification strategies. Prominent examples of diagnostic aptamers. Thrombin aptamers, used in assays to detect and quantify thrombin, which plays a key role in coagulation. Aptamers for cancer biomarkers. Specific aptamers have been developed to detect proteins associated with cancer, such as prostate-specific antigen PSA in prostate cancer. Biosensors. Aptamers can be integrated into biosensor platforms for in vivo monitoring of analytes or biomarkers. By engineering aptamers to recognize specific molecules, they can be incorporated into implantable or wearable devices to continuously monitor levels of metabolites, hormones, or disease biomarkers in real time. This can provide valuable information for disease management or personalized medicine. Binding mechanisms. Aptamers bind to their targets via a combination of non-covalent interactions, such as hydrogen bonding, electrostatic interactions, and van der Waals forces. This specific binding is critical for the functionality of aptasensors, as the binding event is what triggers the sensor's detection mechanism. Design of aptasensors. Aptasensors integrate aptamers as the recognition element. The basic components of an aptasensor include aptamer binding to the analyte. Transducer. Converts the binding event into a measurable signal. Transducers can be optical, electrochemical, or piezoelectric. Types of aptasensors. Electrochemical aptasensors. These sensors measure changes in electrical properties, like current, voltage, or impedance, upon binding of the target to the aptamer. The aptamer may be immobilized on an electrode surface, and the binding of the target affects the electrochemical properties measured. Optical aptasensors utilize changes in optical properties such as fluorescence, absorbance, or surface plasmon resonance to detect the interaction between an aptamer and its target. Fluorescently labeled aptamers are common, where the fluorescent signal changes upon binding. Piezoelectric aptasensors measure changes in mass on a piezoelectric material surface like quartz. The binding of the target to the aptamer alters the mass and consequently the vibrational frequency of the material. Now, modification and stabilization. To enhance performance in biosensors, aptamers may be chemically modified. Common modifications include addition of stabilizing groups such as 2-cetizen, also fluoro or 2-silyene, O-methyl groups to RNA aptamers to enhance nuclease resistance. Labeling with fluorophores or electrochemical tags to facilitate signal transduction. Incorporation of biotin or thiol groups for immobilization on sensor surfaces. Challenges and strategies in aptasensor design. Regeneration and reusability. Designing aptasensors that allow for the easy release and rebinding of the target can enhance the sensor's lifespan and cost-effectiveness. Sensitivity and detection limits. Employing signal amplification strategies such as enzymatic amplification or nanoparticle-based enhancement can improve the sensitivity of aptasensors, selectivity in complex samples. Ensuring that the aptamer does not cross-react with similar substances in complex biological or environmental samples is crucial. Examples of applications. Medical diagnostics. Detecting biomarkers for diseases like cancer or heart disease. Environmental monitoring, detecting pollutants or toxins in water or soil. Food safety. Detecting pathogens or contaminants in food products. Targeted drug delivery. Aptamers can be utilized to enhance the targeted delivery of therapeutic agents to specific cells or tissues. By conjugating aptamers to drug carriers such as nanoparticles or liposomes, they can selectively bind to target cells and facilitate the delivery of drugs to the desired location. This approach improves drug efficacy, reduces off-target effects, and enhances therapeutic outcomes. Nature and synthesis of aptamers. Aptamers are synthetic oligonucleotides, either DNA or RNA, that fold into unique three-dimensional structures, enabling them to bind specifically to a designated target, such as a protein, a specific cell type, or even a virus. They are synthesized using the SELECT, systematic evolution of ligands by exponential enrichment process, which involves iterative rounds of binding, separation, and amplification to select aptamers with high affinity and specificity to the target. Mechanisms of targeting. Aptamers can recognize and bind to specific molecular markers expressed on the surface of certain cells, including diseased cells like cancer cells. This targeting is achieved through molecular interactions that are highly specific, akin to a lock and key mechanism, allowing aptamers to bind with high fidelity to their targets. And drug conjugation and complex formation. Aptamers can be conjugated to drugs or encapsulated in nanoparticles to form drug-aptamer complexes. These complexes can be designed to directly conjugate the drug to the aptamer, where the drug is linked to the aptamer via a covalent bond that can be cleaved upon reaching the target site. Encapsulate the drug in aptamer-guided nanoparticles, where the aptamer serves as a targeting ligand on the nanoparticle surface, enhancing the delivery of the encapsulated drug to the target cells. Controlled release mechanisms. Controlled release is a critical aspect of drug delivery systems to ensure that the drug is released in a sustained manner at the target site. Aptamers can be engineered to undergo conformational changes upon target binding, triggering the release of the drug. Additionally, the linkage between the drug and the aptamer may be designed to be cleavable by specific enzymes or conditions present only in the target microenvironment, ensuring that the drug is released only upon reaching the target. Advantages of aptamer-based drug delivery. Reduce toxicity and side effects by concentrating the drug's action on targeted cells, minimizing exposure to healthy cells. Increased efficacy. Enhancing drug accumulation at the disease site can improve therapeutic outcomes. Versatility. Aptamers can be designed to target a wide variety of cells and conditions. Challenges in development. Stability. Aptamers are susceptible to degradation by nucleases in the bloodstream, requiring chemical modifications for stability. Off-target effects. Despite high specificity, there is always a risk of binding to non-target cells, necessitating thorough testing. Regulatory and manufacturing hurdles. Production of aptamer drug conjugates must meet high purity and safety standards. Pathology. Examples of therapeutic applications. Cancer therapy. Targeting tumor markers such as prostate-specific membrane antigen, PSMA, in prostate cancer or HER2 in breast cancer. Autoimmune diseases. Targeting immune cell subsets or inflammatory cytokines specific to conditions like rheumatoid arthritis or multiple sclerosis. Conclusion. Aptamer therapeutics represent a burgeoning frontier in the realm of precision medicine, offering unparalleled specificity and adaptability in targeting a myriad array of molecular targets. The unique structural flexibility of aptamers allows them to assume intricate three-dimensional shapes, which are crucial for their selective binding to disease markers, be it proteins, small molecules, or even whole cells. This specificity not only enhances the therapeutic efficacy, but also significantly minimizes off-target effects, which are common in traditional drug therapies. The development of aptamers through the SELECT process embodies a powerful platform that has been refined to produce highly optimized therapeutic agents swiftly compared to traditional methods. Furthermore, the chemical versatility of aptamers allows for various modifications, enhancing their stability and circulatory persistence, thereby overcoming some of the main challenges faced by biological therapeutics. However, despite these promising aspects, the path from laboratory to clinic is fraught with challenges, including regulatory hurdles, manufacturing scalability, and ensuring consistent batch purity. Yet, the successful clinical application of aptamers such as pegaptinib and Macugen in treating age-related macular degeneration serves as a testament to their potential, sparking further research and development in this field. As the scientific community continues to unravel the complex interactions between aptamers and their targets, and as technological advancements improve their delivery and stability, aptamer therapeutics are poised to become a cornerstone of targeted treatment strategies. They offer a beacon of hope for myriad conditions, particularly those where conventional therapies have faltered. Moving forward, the continued evolution of aptamer technology will undoubtedly play a pivotal role in shaping the future of personalized medicine, making diseases that are currently difficult to treat more manageable and potentially curable. Stay curious, stay innovative. I'm Luke McLaughlin. If you would like to learn more about these topics, consider reading our full-length articles on www.biotechnologyreviews.com, or consider our podcast series on YouTube or Spotify at our Biotechnology Reviews channel. Our articles are also available on our LinkedIn newsletter,